My good friend Jon Hess is a wonderful human being and a goddamn genius. Ever since I've known him, he's had this ability to be absolutely hilarious and thoughtful. And he's just super intelligent. There was an article that Upstate Medical University just put out about how Jon secured a large grant to help fund some of this research that he's been putting together. Alongside with his colleague and fellow PhD researcher Stephen Glatz, their project BrainGENIE is being developed to find specific gene markers from blood samples and actually brain samples from deceased donors to help identify various biomarkers that would help identify various psychiatric disorders such as schizophrenia, bipolar, and even autism. And Jon explained in the podcast that those specific psychiatric disorders are really hard to, I guess, quantify on paper. I really hope I use that word. That's a word I never use. Really hard to prove, really hard to put on paper. And if you're interested, you could go to the show description below. There'll be a link for the article. I know I myself, not going to lie, I had to read it like 5 times to totally understand what's going on. So I figured after I read the article, I was like, Jon, you should come on and we'll talk about what you've been working on. So it was good catching up with Jon, and I always love conversations with this guy. It goes from really like thoughtful thinking to all of a sudden hilarious, gut-wrenching laughter. I really, Jon is one of the favorite people on this planet. And I love this conversation we have. So here is episode number 43 with my good friend, Jon Hess. And there we go. So I feel like the best way to start this is just kind of introduce yourself, Jon. I know you very well, but
yeah,
people might not.
Yeah, I know we've known each other like 12 years now, right? Maybe more.
High school. 10th grade. I remember sitting next to you in Mrs. Cook's graphic design class with Mark. That was when we first met.
That was the first time. Oh my gosh. Yeah. So yeah, we've been friends for a super long time, and now here we are. So super happy to be on this podcast, man.
Dude, only fitting.
I know. So yeah, a little about myself. So I'm a research scientist. I work at SUNY Upstate as an assistant professor. I'm in the field of psychiatric genomics, and yeah, I'm really into developing different bioinformatic tools to solve kind of prevailing issues in the field of psychiatry.
Why did you go into psychiatry? Like, what was the pull to that particular science?
Yeah, so it was kind of just one of those things of opportunity meets preparation, I guess. Nice. I had a lot of interest in doing something brain related in the field of research. And I was fortunate enough to land a volunteer position in a lab that was doing a lot for psychiatry. With just how things kind of played out, I found a lot of interest in the work that was going on. I think, when I entered this volunteer position, it was right on the precipice of the field of psychiatry taking kind of this paradigm shift, I'd say. And it was really exciting to be a part of that, like witnessing this huge change.
Well, what contributed to the giant shift?
So basically it's kind of this history of labs kind of realizing that there's a pressing need to look deeper at these disorders, which are, if you're familiar with how psychiatric disorders are diagnosed, it's using this DSM manual. You may have heard of the DSM before. It's a diagnostic and statistical manual for mental disorders.
What does it do? Is it like a series of questions, or is it?
Exactly, yeah. So it's basically just an interview-based checklist that clinicians use for determining what a person's symptoms are, and whether or not there's symptoms that they're basically able to determine from an interview standpoint meet up with the criteria for a disorder.
And this is how somebody would get diagnosed with something, right?
Exactly.
Like schizophrenia or something.
So this is where there's some people who have a little bit of skepticism with psychiatry because to them it's perceived as being complete subjective, and that's not totally true. So with psychiatry, clinicians rather are able to reliably diagnose these disorders using simple checklist based items.
And it's just an interview. It's just asking a bunch of questions.
So not something. So these are disorders at present, which you can't simply do like a blood-based test for. So if like you, for instance, had cancer, there's really good diagnostic tools that clinicians can use to determine what type of cancer and staging, the course of disease. That's not
right, and it's through blood testing, right?
Yeah, through blood testing, and that's something that we're constantly trying to push forward to is how can we obtain a laboratory-based objective biomarker for psychiatric disorders. So when I kind of came into the field, this person who was interested in developing a career in science, there was this huge outpouring of data that suddenly came forth through these massive international collaborations where people are pulling together data to study how DNA mutations are contributing to psychiatric disorder risk. And this was something that has been chased in different configurations for the better part of, say, 3 or 4 decades. There's some suspicion going back to the 50s to these family-based studies that were done that made this awareness that disorders of mental illness tend to run in families. So that gives us an idea that genes play a role. But from those very kind of crude studies that are simply looking at how siblings manifest disorders in similarity to one another, doesn't really point to what specific gene or set of genes within DNA.
Yeah, right. And it wasn't like there was no test being run, right? It wasn't just like at the time it was nothing you could test for. Yeah,
the technology wasn't there. It just wasn't available. Molecular biology.
But they still saw that pattern.
Exactly. So they're seeing this pattern, and they're seeing, there's this growing suspicion that genes play an important role. And it wasn't until 50 years later where molecular biology catches up, and now there's technologies available to actually profile the DNA and compare sequences of DNA between huge groups of people, where you could do tests, statistical tests, and see
all with just a blood draw.
From a blood draw, and extracting DNA from that blood, and doing this for tens of thousands of people, some who have a disorder and some who don't.
Right.
And doing these statistical comparisons, mapping out across the entire genome those sets of genes that are starting to show some level of contribution to things like schizophrenia and bipolar disorder depression.
So it was like you just stepped into that field, and all of that data was coming out at once.
All of it was now finally coming out, and there's some really interesting, reliable hits coming. And this was, kind of backstepping just a little bit. So there were these kind of smaller scale pilot studies done using groups of, say, a couple hundred individuals, at most maybe a couple thousand, where there were some clues that maybe 1 or 2 genes could be playing a role, but nothing was really shining out with much confidence. And it became kind of increasingly clear that the clues to what genes in the genome play a role are going to require massive amounts of data from many thousands of people to detect a reliable hit.
Right. Because you need all that other data to like kind of cross-reference, I guess you want to say.
Yeah. So kind of what it boils down to is in psychiatry, these are complex multifactorial disorders, we say. What that means is that there's not one single gene or even a handful of genes that determine one's risk for developing them. It's really a contribution of many hundreds to maybe thousands of genes with very, very small effect sizes in terms of increasing your risk. We're talking about maybe an increase of 1 to 2, maybe 5%. very,
Right.
very small relative to the background risk that you would have if you just randomly selected one person out of a group of 100.
Oh, right, right.
So, yeah, these
Because
are
it's
really
such a
small.
smaller piece of like thing to look for, right?
Yeah, it's really small, and it's pretty clear that it's like the answers are not as scientists would have hoped for. You kind of hope to see a smoking gun whenever you do these big, large, expensive studies of
Right.
maybe one single gene that you can say, okay, this is the gene that's
Without a doubt, this is it. But
gene.
it's a little bit harder with these kind of
Yeah,
psychiatric disorders.
exactly. Um, there's different ways to look at it too. It could just be, it's thought for some time that maybe the reason why we're not finding maybe a smoking gun for, say, depression or schizophrenia is because these disorders kind of encapsulate many different symptoms. No two
Right.
people have to have the same set of symptoms when they're going through a DSM interview that are
Yeah, right.
the disorder. So you have two people with, say, schizophrenia who are vastly different. One can have very complex delusions and grandiosity. The other may have more so the prototypical picture of negative symptoms of schizophrenia, where they become more withdrawn and more catatonic. So those are two very different flavors, but they end up getting lumped together under
Right.
the larger umbrella of a
It
disorder.
makes sense. Like that, all that stuff makes sense because it's when you were talking about that it like runs in the genes. Like one of the things when you go to the doctors, the first thing they ask is like, does your family have any medical history that we need to know about? Right. So it's like if liver cancer can run in the family, then I guess different forms of schizophrenia or depression or what else would you say is also like a psychiatric?
Autism, for instance, is something
Okay.
we study too. Yeah, so there are many different.
Right, that's a good example. That's like a very wide spectrum. Not every
Huge
person
spectrum.
has it differently.
Yeah, huge spectrum, and then even within within psychiatry too. So autism is one rare case where while it does now encapsulate many different forms. When we talk about autism, there's Asperger's, high functioning people with autism, or you have those who are extremely nonverbal. They have a very severe form of autism, who are very developmentally delayed and cognitively delayed. So within that kind of dimensionality space, there are a few cases that can be attributed to, say, one or just a couple of genes that have very, very strong effect sizes, kind of analogous to what we see in cancer. Maybe not to as strong a degree, but maybe you're familiar with like the breast cancer risk gene BRCA. It's one that clinicians are now routinely testing for
because that's a high sign of breast cancer.
It is, and if say like within a family you start seeing signs of a mother having breast cancer who has the BRCA mutation, they often will test then the daughter and maybe the grandchildren too, to see if they carry that same gene, because that increases your risk for breast cancer many-fold.
Wow, that's crazy.
Some of that kind of that flavoring gets picked up too within the realm of psychiatry for some of those developmental disorders like autism. But disorders that occur typically later in life, or at least detected later in life, like schizophrenia, the mutations there that are contributing to risk are much more subtle. And that kind of brings us back down to this prevailing challenge within the field is how can we go about better diagnosing these disorders? How can we better understand the biology of these disorders in the brain? Because currently we have the ability to generate massive amounts of data for these disorders, whether it be brain-related data or data simply from blood, or data about the genome, or different facets of molecular biology like the epigenome. But putting all this information together to get a clear answer is very difficult.
It's been very so. When you like first approach this problem, was it always like to do a blood sample, or like you knew like you would have to get something from a sample, right? And then was it maybe we broke it down even farther?
Yeah, so to kind of go through kind of like the chronology of this and the motivation leading up to this new work that I'm involved in developing BrainGENIE. My lab is one where we tend to focus a lot on the accessibility of blood in terms of identifying useful sets of biomarkers for mental illness. And the reason why we look at blood is, well, A, it's very easy to access. It's a simple blood draw.
And you can get a lot of information from it.
Yeah, and you can get a lot of information. There's a lot of genes expressed in blood that are also expressed in brain. So there's some good empirical data that have been done through different studies of humans and of non-human model organisms to show that there are many sets of genes that are highly expressed in blood that are also very highly expressed in brain. Not all genes by any means. There's definitely some very brain-specific genes that we can't find in blood,
right,
and vice versa. But
so you just have you have the blood readily available to test. So that was like a good place for you guys to pretty much start from.
Exactly, because when you're talking about patients who are alive, we can't very well dig into their skull and take out brain tissue sample. It's unreasonable, it's impractical, and there's
when somebody donates their body, that's part of what their brain goes to, is maybe a study like this.
Exactly, yeah. So there are very few cases in which we have the ability to look at brain tissue within the space of psychiatry. Typically we're talking about people with schizophrenia, it's post-mortem brain tissue. So this is after they've died and their bodies have been donated to science. And we're able to look at brain tissue basically after death and make some inferences about how genes at that point in time may relate to the person's condition.
Do you think the tests from the brain produce more accurate results or more consistent results?
So in some ways, it's believed that if we're studying the brain, where we believe that's where the disorder should initiate from, they should originate from the brain because they're brain-related phenotypes, that we should have a clearer answer.
Wait, did you say genotype?
Phenotype.
What is that?
Like the manifestation, basically. Okay. In this case, any sort of type of trait.
Okay, I gotcha.
Yeah, so if it's a brain phenotype, then naturally we should look at the tissue of origin of where that's actually manifesting. Right. And it's not like in brain cancer where you can target a tumor or a set of tumor cells within the brain. We don't know really where schizophrenia exists because it's basically locked into sets of dysregulated or malfunctioning synapses between sets of neurons.
Right.
Or circuitry.
So much harder. Yeah, I never thought about that. You're so right. It's like you could see a tumor, but when it's all when it's a mental and it's a way that the brain is functioning and firing off on cylinders, that's what makes it hard to put data on paper.
Extremely challenging. Yeah. Right. So we know that if there's certain neurotransmitters that may be imbalanced in people with schizophrenia, for instance, and we know even from neuroimaging studies that have been done in thousands and thousands of individuals that there are subtle brain size differences that do occur on average in the population of people with schizophrenia, but that's not enough evidence from those large group-wise comparisons for us to say do a neuroimaging test on someone to see if their brain has changed. Because that in itself is not a very good marker for these disorders. So that we do have kind of some evidence that we can glean from, say, post-mortem data where we can carve out tissue regions from all over the brain and run our experimental tests on those tissues to see what genes are maybe active or altered in some way or maybe not expressed when we expect them to be expressed. And then we can, combine that with information we've seen from, say, neuroimaging studies to kind of correlate those two, and even connect it to even further to all different modalities of these large experiments that are done. But still, there remains this pressing question of what can we actually do to better understand these disorders when people are alive. So it doesn't really help us too much to describe the conditions after death.
Yeah, right. But trying to find the link.
We're talking about people with these disorders for typically decades. So we're talking about when we actually do these postmortem studies, we're kind of hindered in some way by the fact that whatever information we get out of the brain tells us basically a portrait of what it looks like to have schizophrenia at the molecular level, if you've had the disorder for many years and you've been treated typically with a hefty regimen of antipsychotics and a whole host of other neurotropic medicines.
Right.
So there's a lot of these factors that come into play, which make the results a little bit more perplexing at times, and tend to lead to more questions than they do good answers. So that's what brings us to...
That's how you started to develop BrainGENIE, right?
Exactly. So
then did you develop that with your because you're the assistant professor? Who's the other person that works with you?
So the other person I'm working with closely on this tool is Dr. Stephen Glad. He's someone who I started off at Upstate with, and as a volunteer. I did my doctoral dissertation with him, and I even did postdoc training. So we've been working together for a long time, almost a decade. Yeah. So we developed this tool together to utilize blood data from a large cohort of individuals who were collected as part of this large NIH-funded study that was started, I think, back in 2013 or 2015 is when.
And what was it again?
It was an NIH study funding. It's called a Genotype Tissue Expression Project, or GTEx.
What was that? What do the abbreviations stand for? Do you know?
For NIH, that's the National Institute of Health.
Oh, okay.
Our main funding sponsor for most studies that are done in the U.S.
That was going to be one of my questions, because it's like, for somebody who doesn't know that level, is like, how does somebody in your position typically secure funds to do this kind of research? Is it through donors? Is it through, like, how does that system totally work?
Yeah, so it's typically the NIH would be the go-to if you're an academic research center, like Upstate, where the majority of people who are doing research are getting their funding through independent means. NIH.
Do you have to still do like remember in high school the scientific theory? Would you have to do a scientific theory outline and say, please fund my research because X, Y, Z?
So there's a pretty hefty protocol involved in terms of how you go about getting grant money through a funding agency, and they're pretty similar. So NIH has sort of their own standards, and they vary across different funding mechanisms. So the specific project that I'm working out for BrainGENIE that was funded by the NIH, that's through what's called an R21 mechanism, and that one is a 2-year funding cycle. And what it's kind of intended to be is a way for a study which typically has some preliminary data and has some feasibility behind it to get the necessary resources in place so that it can be taken basically to the next level, so that it can then be in an even better place to secure even bigger sets of funding.
Right. So
usually,
I always imagine it's like when you guys secure funding, it's like a giant gala. You guys just throw a big old party and rich people are like, this sounds cool. Here's a lot of money. But I think that's my weird fantasy brain.
So I wish it was like that. It's actually not as...
Holy cow, you look great in those glasses. Here's $2,000 for your research.
That would be remarkable. So no, it's actually a much more painstaking process.
It has to be.
Yeah, so it's the application cycles. They occur at different increments throughout the year, depending on what mechanism you're targeting through the NIH. So for our R21 grant, we applied for that initially back in 2020. I think it was in September, and then several months goes by. You just kind of wait and see how it goes. From there, the grant application will go to a scientific review committee, and that's made up of a large set of reviewers, people who are actively doing science, many of which have their own funding. These aren't necessarily people who work specifically for the NIH. This could be researchers who are recruited to come for a set of days to review many grants and then determine from this set of grants which are most likely to be successful and be innovative and significant and worth the investment. Because
if you secure the funds, do you have to meet all these hurdles and markers basically?
Yeah, so there's basically a huge set of criteria that you're evaluated on. when you apply for these grants. So again, it's a very competitive atmosphere. It's basically a numbers game. You have to, the expectation is if you're an academic researcher that you're most likely not going to get your first grant funded. And this is not the first grant I've applied for by any means. There's been many in the past. So there's a variety of reasons why a grant can fail. It could be for something as trivial as formatting issues, which would get you
like you formatted the letter wrong.
Yeah, if you formatted,
oh Jesus,
say things like your BioSketch, for instance, which is sort of like the snapshot of like you as a researcher in your career. That's something that's updated pretty routinely. I don't want to say
it's like how dare you use Arial font? You should use 10 point font. You should only use Arial. Make sure the margins are 1.5 inches.
Exactly. So it's quite stressful because like it's something as important as like the formatting can mean success or death within this.
And then if they say, oh my God, have the margins are 1 inch more than they should be, and they kick you back, like you start all over again with a new. If you
take up more space than you're allotted, that's it. Usually, it's
God,
kind of a quick way of triaging grants would be my guess because they're getting inundated with so many. There has to be some easy ways to determine off the bat of this huge stack of grant applications who can stay and who we can actually just kind of set aside.
So they really set the bar high on those kind of things then.
Yeah. So not beyond the trivial stuff of just formatting, there's sets of criteria that are evaluated within each grant application. And they're relatively fixed for the most part. But some are weighted a little bit more heavily depending on which mechanism you're applying for. So kind of broadly speaking, there are grant applications that are maybe more so tailored to junior trainee, someone who has graduated with their PhD, but they're not yet an independent scientist. We call these usually postdoctoral fellows or postdoctoral associates. So it's someone who's ongoing additional training beyond their PhD years in preparation for, say, applying for a faculty position. And there are specific training grants available to the postdocs. So in that sphere, it would be very important for a postdoctoral applicant to demonstrate that they have a really good training potential and a really adequate training environment, that their supervisors have a strong track record of, say, publishing in a specific area in which they're going to receive that training.
Right.
So that would be, one major piece of criteria for that grant. But that's not something that exists in someone who's, say, a senior scientist who's been in the game for 40 years, because they're obviously not needing to seek out additional training unless it's very specific. Usually in those spheres, you would be evaluating that senior candidate for a grant on.
Wow, that's crazy. What a process.
Yeah, you'd be evaluating for things like how impactful the science is, the potential for it to shape the field. The novelty usually is something that's increasingly given more weight. And by novelty, I mean, is the thing you're doing either brand new, has it never been done before, or are you doing something that exists in a new way?
Right.
So
this has got to be brand new. Like nobody's ever done something like this, right?
So I wish I could say that. In the honest way is that there's a lot of people who are thinking about this, but very few labs who have actually distilled down this type of thing to practice. And of the few labs that are actively working on this type of what we're calling brain region specific gene expression imputation, we can break that down later.
Dude, that's a freaking mouthful right there.
So of the people who are attempting to do that, no one has really come as close as we to having as comprehensive and accurate a method. So
wow, that's still impressive though.
Yeah. So that's, it's with anything. If you have a good idea, there's a high likelihood that many other people in your field have that same idea.
Right.
And you're always kind of playing the game of catch up in some ways. It's like who has the ability to kind of push that work forward quickly enough so that you don't get scooped. In our case, I don't think we've gotten scooped. In fact, I think we're fortunately a step ahead in many ways.
Wow. That's cool. So has it just been you and that other doctor that
Yeah,
you work with?
and then we have, there as with anything in this level of science, we the success of like a method like this is based heavily on the availability of data that we have to kind of develop the method and validate its performance. So there's a couple of other labs that we collaborate with. So on the paper itself that we're working on right now to actually publish the method, we have about a dozen or so individuals who are part of that group, who have helped in some way in terms of either contributing data or just kind of supporting the conceptualization of the tool itself. But yeah, it's basically myself who's the main charge in terms of I would say the kind of the engineer behind the method, actually writing the code to develop it and make it work.
Oh wow, that's
yeah,
crazy.
so, yeah,
That's insane. And it's like it's so interesting. So it's cool because when you say you're working with all these other offices and labs and stuff, I kind of think of it as almost like an open source video game, right? Like Skyrim. Like Skyrim is a great game, but then you have all these cool add-ons that other people made. So you have like all more data, right? And the same thing for the lab is like you have this great like database, right? And then you are adding everybody else's database to make this thing only bigger and only quantify the results even better, right? Like even set that stuff in
the way that we advance science in this era right now is like no single lab is able to really exist on an island and be able to kind of be a powerhouse.
Yeah.
The kinds of questions that are being dealt with require access to huge amounts of data, which are typically extremely expensive to generate. And there's a lot of data that have already been generated by hundreds of labs. So it doesn't make sense for these smaller data sets to exist in these smaller scale labs. It makes more sense for people to be pooling all their data together into a resource so that scientists all over can access the data, analyze it, and make
it's like this sense of community.
Yeah, that's the huge drive. And also kind of, again, why I decided to stick in psychiatry is just this kind of ethos within this space of kind of teamwork and collaboration and the need to continue striving to push for open access to data and open source tools like BrainGENIE. So that's one where we've, we're not seeking out like a patent on it at this stage. In fact, the tool already exists. It's already out on the internet. People can download it and start running it if they wanted to. And it's the kind
just
of
install the add-on. You install the add-on, then you can ride the dragons and ride the horses and stuff.
Yeah, so that's the hope is that a lot of people are going to be using this tool and making fascinating discoveries with it, even beyond what I'm able to do with it. But
that's cool.
Yeah, I'm excited because we do have a tool. It's been one I've actually been working on it for the better part of 3 years.
How do you code? Like, how are you designing? Now we get into I guess the technology and software.
Yeah, like nuts and bolts.
You're like writing a code for a program to run these series of tests. How did you start? How did you start that project? I guess is what I want to ask.
Yeah. So really the existence of this project is really in large part thanks to the availability of this huge resource that was developed by this GTEx consortium, the one that was funded by the NIH to basically map out molecular profiles from people who donated their bodies to science, hundreds and hundreds of people. So this resource itself, it's kind of grown in size and there's been different iterations of it. And we were fortunate enough to be able to access this resource, download the data and start kind of toying around with it and seeing what the capabilities of this resource really are.
That's cool.
So they're kind of in the midst of this, we became aware of a few other labs who are working on utilizing kind of a different entry point to the brain in terms of making predictions about gene expression profiles of
like if somebody would have a higher chance of getting schizophrenia when they're over 60, right?
Yeah. So these are kind of the tools themselves are meant to be kind of generalizable enough so that they can be kind of used to make inferences about a whole host of different brain-related conditions and phenotypes.
Right.
Ours specifically, we're not just interested in schizophrenia per se, or just brain disorders. We're trying to basically just understand very fundamental questions about how the brain works, how it develops, how it matures, and then what happens to it when something's gone awry and there's a disease forming. So we a couple of years ago, we became aware of these other tools that were, attempting to take a crack at predicting what's going on in the brain. And we saw an opportunity within the space to use our expertise about the blood and its comparability to the brain in terms of the proportion of genes that both tissues express, to develop some new models where we could theoretically take a blood sample from someone, profile information about their blood to see which genes are expressed,
and basically just run it through a system.
Yeah, run it through a computer system that has basically a machine learning model built into it.
And so that's what you're putting together?
Yeah, it's basically kind of, and fundamentally, it's just basically a prediction model. It's using input about the blood to make predictions about the brain.
Cut to the Jon Hess 1980s montage of you just hitting zeros and ones into a green and black monitor.
Yeah.
Just all different views, and all you're just doing is going 111000011111000.
Just like all this like code falling down on screens or 10 all around. Yeah.
What program are you using to write like the code for it?
So the one I'm using is called R. It's a tool that was developed, I think, like 30 years ago by some biostatisticians.
Whoa.
It's not super common within the field of, it's one that's becoming increasingly popular, but more so among people who are doing data science or people who are doing bioinformatics. But if you're, say, a hardcore engineer working at Facebook or Google, it's unlikely that it's one that you would use. You'd be probably using something much more lower level for computer programming, like I don't know, C++, C#, Java,
right?
Or maybe something like Python, which is.
Is it because there's just so much data going into this? Is that why you would use?
Well, so R was one I started off programming in. So obviously I didn't have any type of formal training in programming in college. When I joined the lab, I really didn't know how to program at all. I could do basic stuff like develop macros in Excel. But for the size of the data that we work with, it was becoming increasingly clear to me that I needed to work with something that I could start to automate some processes and have control over huge sets of data in a reliable way. So then I became aware of this tool R, and took a liking to it, specifically because it's kind of intuitive if you're from a statistical background and you're trying to figure out a program. A lot of the routines that are available to you within R kind of make a lot of sense. So it has a kind of a base suite of things that would allow you to run a lot of different statistical tests.
So this is all self-taught, John?
Oh yeah, it's all self-taught.
Jesus, man.
Yeah, it took me a better part of like a year and a half, and I still feel like I'm constantly learning tricks and neat.
Is that what you did in like when COVID hit? You're just like, I'll just learn how to freaking learn to code.
Well, so actually kind of going back further than that, when I started my PhD.
Oh, wow.
Yeah. So part of my PhD was I wanted to analyze, these huge sets of data. And before even kind of starting my didactic learning, I was kind of teaching myself like off hours how to program. So it took me about a year.
God, dude, you're a boss.
Kind of proficiency with it.
We always knew you'd be the smartest one. We always knew.
Thanks, man. I really appreciate it. But yeah, so it's self-taught, but it required kind of like team effort. There was some fellow PhD students too who I was working alongside in lab, and we were kind of both struggling through these steps, like trying to program in R for the first time. So we would kind of bounce ideas off each other and troubleshoot, and it was kind of a fun, clunky process. Made a lot of mistakes along the way.
There probably is not very like many YouTube tutorial hows on R programming, is there?
It's hard. It was hard to come by. Yeah. I mean, now you could probably watch hours and hours of stuff.
Really?
But really, in terms of like, you can really only learn a language, I think, in terms of like project-driven goals, at least for me. So I tried doing stuff like these, like Code Academy, like free open source things online where you can.
Oh my God. Dude, this is gibberish.
yeah, like learn how to code by typing in print and hello world in quotes, like some very basic rudimentary stuff like that.
Is
Yeah,
this it right
exactly.
here?
Yeah, that's exactly what it looks like.
It's just a bunch of like this is like remember when you would make a MySpace layout?
Yeah,
you
yeah,
were like coding all of this
HTTP
stuff.
and stuff. Yeah, so there's a lot of cool functionality to it, and it has a lot of similarities with Python, if that's one you've heard of. It's another really super common tool that data scientists are using. Python has a really good rep right now for its availability to do some complex machine learning, like deep learning type things for image recognition, and audio processing. And R is sort of catching up to Python behind it.
Interesting.
Yeah, so the two are kind of going back and forth. There's like this ongoing debate in the field, which is kind of like when people are nerding out, like which is the better tool for solving X problem in bioinformatics, Python or R. And really, at the end of the day, it doesn't matter. If you're good at one, just stick with that.
Yankees or the Red Sox?
I know, it's like this silliness. Like they both kind of, in the end of the day, do the same thing. You just write in a different way. So
and I'm totally fixing my MySpace layout when I get done with this. That's essentially what it is. That's what it looks like. It's just like a bunch of like parentheses, dashes, all this other stuff. Man, does it like make a unit like a interface or something after you get done coding? Like, the MySpace layout, I keep reverting back to it, but you would put code in and then you'd go to the page and it would actually look like you did something. You know what I mean? Yeah,
like it would kind of like compile it and you would get like kind of like a visual layout.
Right.
So you kind of yes and no. There do exist some tools within R, like add-ons. You can like download them. So like people who are much smarter and like who are like legit programmers have developed these add-on tools for R where you can launch applications from R, and you can have servers run these things on the web so that people can actually visualize functions that are being run on data without having to actually write code out. It's not very good though for, say, like you can develop games with Python, but I don't know it's theoretically you could develop like a visual game with R, but it would be very clunky.
It'd be like Myst.
Yeah, like
not very many animations. It's a puzzle game.
Yeah, so like obviously you would want to write a game in something like C++ because that's a language where you're kind of going through fewer obstacles for the computer to run the code.
Oh,
so
right.
Something like R, it's considered like a high level scripting language. So when you run a command, it's like R exists sort of in the foreground of the computer space. Like you as a user, you're kind of interacting with this tool, which is then in the background talking to the computer through several layers of code to get down to the machine code. So it's a lot slower in terms of the way in which you can interact with data. But
that language wasn't made for video games. That was made for you had a term for it. Study science or like
so for data science, because it's like you want to be able to solve problems quickly. You don't want to be sitting there trying to get a compiler to work correctly,
right,
and not be thrown off by the addition of some indent in your code.
Yeah, you're not trying to play like Dust on Counter-Strike.
Unless you're developing like a super specific tool that's churning out huge output on massive data sets, talking like gigabytes or terabytes worth of data. Like there's then like
how much data does somebody's like brain tissue reading take up?
So it can be quite large. If we're talking about sort of like the analysis ready data set that I would be interacting with for BrainGENIE, it can be condensed down to a relatively small space of maybe like a gigabyte or so. So it's not huge by any means, but it's also not small. Like it's too large for you to be interacting with it in the space of like Excel, but not by any means like the largest scale data that we typically would be seeing churned out nowadays.
Right.
There's massive data sets that now exist about the genome from these huge sequencing studies that take up like terabytes or petabytes worth of space on a hard drive. So there's this.
And are you also finding like not only just these? Are you finding? I think you called it biomarkers for other things that aren't just like mental disorders, like being a higher risk of having lung cancer, like we were talking earlier? Are you finding markers like that too?
So there, in my work itself, no, I'm not really seeking that out just yet. I mean, there's
because you focus on just the brain, right? Like
that's kind of from like, from the chin up, or if you want to call it, that's kind of where my expertise lies. So that's what I usually focus most of my efforts on. But the tool itself, if we're talking BrainGENIE, the guts of it are very generalizable. There's no reason why it can't be done for other organs or tissues within the body.
Right.
In terms of, the brain is not unique in the sense that there are many other tissues that you wouldn't just want to be prodding and taking biopsies of because
right, because the brain is so crucial. And that's one of the hardest things too, is like with anything in the brain is so hard to study. It's like one of the biggest reasons why we have such a hard time studying dreams. You know what I mean? It's hard to get quantifiable data on paper,
Exactly. Yeah. So for establishing causality is like one of the toughest things within our field. When you're talking about how a gene actually influences a disorder. So like when we tend to do these large, what we call observational cross-sectional studies, because they're very relatively inexpensive to do compared to the type of study design where you would get much more information, more direct answers, which would be a longitudinal study. And what BrainGENIE is seeking to allow us to do is to perform longitudinal studies of the brain in people who are alive without ever having to actually biopsy their brain. We could just theoretically take blood samples from people, routinely, or as symptoms start to emerge that might warrant some sort of, clinical insight as to what's going on.
Right.
If we could do that, then we can actually start to develop more clues and more insights as about like what genes are going to be dysregulated as a disorder is starting to formulate in the early stages. And that's, one of the main questions we have is like what happens in early forms of schizophrenia before they become chronic. And it's really hard to access those types of people. Usually once someone gets diagnosed, they're
usually in a severe stage, right?
They're a severe stage, or they could be put on medications right off the get-go. And things like that we have to combat with is how can we get someone who's unmedicated, who's in the early stages of psychosis, and what's going on in the brain that triggers those events, or even events leading up to that. So that's what we're really trying to target. But, there's a whole host of other disorders outside the brain that we could be doing this type of work for too.
That's interesting. That's pretty big. Like that is because I guess if the science can be applied to other parts of the body from like you said, the neck down, right? It would be this very similar process, I would imagine. Still blood sample, still testing for all these triggers and markers, Yeah,
exactly. Yeah, so really it's just a limit of, time. Time is the ultimate limiting factor in science.
Yeah, and focusing on so much, like even just the brain alone is time consuming, but then you have all these other parts of the body, like then the neck down, right? That's a whole nother field of medicine,
It is, yeah, and it requires, a lot of people who have expertise in those different tissues and organ systems and the disorders that relate to those tissues to have insight as to what's going on. So, we could theoretically just kind of generalize this tool out so that we can make predictions about the entire body, about from a single blood draw. And it's a matter of doing it in a way where we can come up with some real biological inferences at the end of the day. So kind of bringing it back to what BrainGENIE is going to be doing as part of the grant itself is we have sort of this tool that exists already in kind of a rudimentary form, but still it's performing quite well, better than really what we had hoped for at this point. But we're gonna try to refine it a little bit more and incorporate some new information, new kind of more sophisticated data modeling approaches into the kind of the nuts and bolts of it.
And is that a program that you're also writing?
Yeah, so we're trying to basically fold into the existing program some more deep learning type mechanisms for kind of modeling the data in a way where we can get some more insights. So yeah, there's some more programmatic stuff that needs to be built into BrainGENIE at this point. But then beyond the kind of the programmatic engineering stuff, we're seeking to apply it to this fairly sizable collection of blood data that we've collated from. Well, some of it's generated in-house through our lab, through smaller scale studies of schizophrenia and autism. But we've also collated data through collaborations, as well as downloaded tons of data through these open source repositories that the government controls. And usually as part of NIH grants, it's a stipulation that once the grant finishes, if you've generated data, you have to then share it with the community, which works.
Oh, I kind of like that.
We can then collect those data kind of after the fact and incorporate it into our models. So yeah, we have this collection around like 8,000 or so individuals where we have.
I love to hear that. I love to hear like that the medical field is not like, no, this is mine. You can't have it. You know what I mean? Like I love to hear that it's just open source sharing with everybody. I love that.
Yeah, it comes as kind of a group effort in this initiative. I mean, years and years ago, that was kind of the mentality because it was so hard to collate that data from the get-go, getting the grant, getting the people recruited to your study, and then generating the data. It's very effortful. And, part of the system is that you could make a career out of a expensive data set, keep publishing on it, making new discoveries. So the incentive was there to kind of protect your data because it was a way for you to protect your career. And it also to support a lab with many personnel and many trainees. Right. But that system, started to dissolve as it became quite clear that there's only so much information you can glean from smaller scale studies. You need many, many hundreds, thousands, tens of thousands of people to really.
So it's like you scratch their back, they're scratching yours. It's a good give and take.
Yeah. So that's the model is such that now, no one lab can possibly generate enough data to solve these complex disorders. So we have to pool our resources together to make reliable discoveries.
Wow. Damn, that's so cool. It's so impressive because one of my favorite quotes is "Thank God for people smarter than me." But seriously, because this is like groundbreaking because this could be very important to really treating people in need.
Yeah, I mean that's really the hope. So once we go about more of the work of the grant itself and in applying BrainGENIE to this collection of data we have, what we hope to accomplish from is identify some sets of genes or some biological pathways that are altered in the brain that we can then map onto these disorders that exist within our collection. So we're looking not just at schizophrenia from our collection of 8,000 people. We have data on bipolar disorder, major depression, autism, PTSD.
And this is the major focus, right? Like that is the focus of this grant, the main objective.
Yeah. So, I mean, like even if say like the engineering component of this grant, it doesn't pan out for whatever reason, maybe we just can't bump up the accuracy of our models. We still have an existing model to fall back on that we can readily apply to the data we've collated. And from that, make some really cool discoveries. So with that, we're hoping to, have basically an atlas of genes that we can map onto disorders. And then from those,
you're building a giant Wikipedia page.
That's kind of the hope. Yeah.
Yeah. That's what it sounds.
This kind of like gangbusters resource that people can access and make, additional models off of. But what I'm hoping for is that we can have from it a set of biomarkers that we can start developing classification models, like diagnostic models, so that if someone were to come to a clinical setting that a psychiatrist has some suspicions about, we could run a panel of biomarkers on that person and see, okay, we're say 80% confident this person meets the criteria biologically for having disorder X.
That was gonna be my next question, was like, so you do this grant, you're able to be successful in building this program. It's like, where do you go from there? What's the next steps? What do you hope to implement in something like that where somebody could do a blood draw and find if they have all the early signs? That's the end all for the BrainGENIE.
That's one of the goals. I mean, we have this very long haul vision as to like how we can see this tool, this technology being integrated into different facets of biological research and medicine.
And are you thinking 5 years, 10, 20, 30 years down the road? Like, how far is the timeline?
Oh my God. I mean, it's at this point,
Hard
it feels
to say.
infinite. Yeah. It's a matter of
right.
like opportunity, technology, access to resources. There's a lot that I would say we could aim to accomplish on our end from just the efforts that we have in place. But there's a lot of stuff that can only be made possible with the right connections, the right tools, and the right kind of buy-in from different institutions. So like, for instance, I think it'd be really advantageous if at some point this type of tool could be integrated into, say, like electronic health record monitoring. So like electronic health records are kind of becoming the staple of clinical practice now. Like your medical orders and your histories when you go see the doctor, they don't exist on paper form. They're being uploaded.
Yeah, not anymore, right?
It's electronic system. So even things like when you go to the clinic and you maybe have a blood test done, like all that information that then gets logged into an electronic database.
Wow, that'd be crazy. Well, this way you can find all these these crazy markers ahead of time.
Yeah, so like kind of integrating that electronic health record initiative with a separate effort, which is biobanking. There's this increasing push within different countries to collect as many biospecimens from just regular patients and see how those biospecimens can be linked with medical information to make discoveries about disease and health. There's this really super awesome resource that just came online a couple of years ago from the UK called the UK Biobank. It's one of the biggest to date, where they've profiled now pretty much like every facet of biology, of like medical histories and biological data from 500,000 Britons. And they've released the data. So we've actually gotten access to that resource, which makes me
Did you put it into the database, or does that information not really apply?
So the hope is that the information could apply. We're currently accessing it for a separate project, but it's kind of inspiring to me because I could see a future BrainGENIE where if the right type of data does exist for, say, a collection the size of the UK Biobank, we could just make predictions off of the samples that are available to us and then link it with the thousands of different conditions that have been tested for.
Right.
So, yeah.
Wow, that's crazy. And then you're juggling other projects too? You said another project you're working on. How many projects are you guys juggling?
So of funded projects, I'm currently on 3.
Wow.
And then within those, there's always kind of these sub projects that you're always experimenting within science to try to develop new leads and new possibilities for new grant opportunities. So there's always lots of kind of irons in the fire. And there's the 3 main funded projects and then several other projects that are kind of supporting that work in different ways, whether it be methods development. So like trying to develop new tools to better the work that's being funded, or to do work that's not strictly funded right now, but could potentially lead to fundable opportunities in the future.
Right.
So yeah, there's always stuff. Lots of collaborations. It's usually a backlog of work, and that's my case. That's just my life. There's always more work than there is time.
Yeah, right. And this is probably a stupid question, but you get into work one day. How do you know where to start? Like, how do you know what project you're working on? Is it just like you're just putting out like a fire over here, you get an email over here, you need to go over there? Like, how do you plan your day?
Yeah, it's tough. Time management is a thing that I'm always struggling with. It's like a lifelong struggle for everyone in this type of field.
Yeah.
Especially for, someone doing what we do, which is like you work at a dry lab, meaning you're in front of a computer all day.
What does a dry lab mean?
So dry lab is this term that's come out to describe people who are doing computational work. So
okay, gotcha.
As opposed to what they say a wet lab, where you're kind of bound to a bench and you're working with like pipettes and test tubes and running kind of experiments in a petri dish, or
exactly like mad scientist style.
Yeah, so like when you think of a scientist who's like wearing the lab coat with like the safety,
we know you're making creepy crawlies in that oven.
Yeah, that's like not my life at all. My life is either
coding in front of a computer.
It's been in my home office in front of my laptop, just coding and
right.
reading papers and trying to write manuscripts and apply for grants.
Oh my God, that's crazy.
Yeah, so I guess there's always that rough there's the growing list of to-dos, and then the day happens, and then that to-do list gets usually shuffled around many times. So you are kind of always having to monitor your emails to see like what fires are needing to be put out. And the hope is always that you're making steady progress on multiple things simultaneously. So it'll
help the research progress faster.
Yeah, it comes down to like self-discipline and time management. And everyone has a different system. I'm constantly writing to-do lists to like by hand. I'll write something for the day. Like I want to do A, B, and C, and maybe I only tackle A like 50%, and the rest are untouched. But at least there's that constant reminder of these are the things that are priorities.
Yeah, too. And then when you write it down, it makes that connection in your brain too.
Yeah, exactly. Yeah, because I have electronic to-do lists and things I store on my notes app on my computer of just these like random ideas of
those never click with me. So much easier for me just to delete them than I do with a written to-do list.
Yeah, I know. At least with like the hand stuff, it feels like accountability. Once it becomes digital, it's almost like it sits within.
There's a disconnect.
Like you can easily ignore it. It's just in the background.
Yep.
It's just like lost now to like this background noise. But
what I'll do too is I'll put the link to the thing that generated this whole thing was this article that Upstate put out about the whole grant that you landed. So I'll put the link for that article so people want to learn more about like what you're doing, and it even goes into a little bit about your background too, how long you've been doing it for. That's sweet. Well, cool, dude. Do you have anything else that you want to share about BrainGENIE?
Just stay tuned. There's gonna be some cool stuff.
Follow BrainGENIE on the gram, baby.
I know it'd be funny to do that.
Dude, I'm excited to see how this turns out. What kind of timeline? Like, is there an expectancy that you're gonna finish it next year, and then you can come back on and talk about what the finished product looks like, or is it gonna be a much longer timeline?
So, I mean, like we have like a manuscript we're working on right now. It's actually in the inboxes of a bunch of our co-authors, and we're hoping to have that submitted to a journal within like a couple weeks. And usually the publication timeline can be anywhere from like 3 months to like 8, depending on if the journal likes it, and if you get like favorable reviews and you're invited to like resubmit. Usually you have to make corrections. So that's usually the first log jam. After that, the full project timeline is 2 years. So that's kind of that's what we're kind of committed to, is having all the work wrapped up within that 2-year space. But there'll be kind of incremental steps. Like we're not just going to have 2 years to do the work and then try to publish it. Like at the end of the second year, we're going to be kind of trying to routinely publish findings as they come out. Every 4 months or 6 months or so.
Right.
So there'll be stuff hopefully steadily coming out in the next like year and a half that I can talk more about.
Yeah, sick. Yeah, dude, you're more than welcome anytime. This was a lot of fun. Thanks for doing it. I learned a lot, and I feel like you did a great job dumbing it down for my Neanderthal brain.
I appreciate it, man.